Just hit an absolute suspicious leg day. Now it's time to take on the beast. So we're going to be doing exam questions. This is mainly going to be on the topics of carbohydrates, biochemical tests, a little bit of water, and it's going to be testing your knowledge on general biochemical mathematical skills. So it's not too bad, but I believe these kind of things are necessary if you want to get good grades. So lock in and let's get this done, bro. So this exam is going to be out of 30 marks. So what I want you to do is count up each mark you get in this exam, and I'm going to grade you later. If you want to be like super nerdy and like, you know, a bit silly about this, you can set a time limit to really emulate a real exam. Set a time limit for about 30 minutes while you do these questions, alright? I will be linking the exam papers that I got these questions from on the AQA tests, but they're a bit muddled because I took bits and pieces around to kind of match what we went through. Alright, bro, enough faffing. Let's get the first question done. Alright, bro, starting off nice and easy. It says here, my G, can you tell me what the bloody bollocks is a monomer? So, pretty easy, I'll give you 3 seconds. 3, 2, 1. Alright, bro. Should have got your answer by now. We did go through this, bro. It's simply a smaller repeating unit that makes up a larger polymer. So if you got anything along the lines of this, or what I said, then that's a 1 mark, bro. Alright, bro. Numero 2. Says here, yes, my G, name the type of chemical bond that joins 2 monomers that form maltose. So, again, I'll give you 3 seconds to get this one done. 3, 2, 1. Alright, bro. So this question's got nothing to do with maltose or anything. This is just basically saying, yo, bro, what is the bond that is made to join 2 monomers together, 2 monosaccharides together? And the answer is a glycosidic bond. So if you get that, bro, good job, man. Well done. You better have gotten that one right, bro. Now, let's get into the next question, bro. Alright, bro, we've got a little bit of a story. So, lactulose is a disaccharide formed from 1 molecule of galactose and 1 molecule of fructose. Other than being a disaccharide, give 1 similarity and 1 difference between the structures of lactulose and lactose. So, I'll just pause the video, get this one locked, and I'll go through the answer in a second. So with this, you could have said many things, okay? So, 1 similarity you could have said is that one thing that all disaccharides have in common that monosaccharides don't is that they are joined together with a glycosidic bond. So that is a similarity that they both have. You could have also said that they both contain galactose, which in one of my videos, I did say you have to memorize what makes lactose, and what it is glucose and galactose joined together to create the disaccharide lactose. So if you got either one of those for your similarity, that's 1 mark. Now for the difference, this is mainly testing your memorization of what makes what. So remember, lactose. To make lactose, you need glucose and galactose. Whereas to make lactulose, you need galactose and fructose. So instantly there you can see your difference. Alright, bro. So if you put your similarity and difference, that's 2 marks. Well done, bro. Next question then, bro. Alright, bro. So we have here a little bit of a Who Wants to Be a Millionaire. So, it says here, yo, my bro, which test identifies the presence of starch? Is it A, acid hydrolysis test? Is it B, Benedict's test? Is it C, emulsion test? D, iodine and potassium test? Or E, I don't know, bro, leave me alone. Alright, bro, get your answer in 5, 4, 3, 2, 1. So the answer is D. Well done, bro. If you got D, you got 1 mark, bro. Well done. Alright, bro. Next question says, explain the difference between these structures. And on one, we got starch, and on the other, we've got cellulose. So pause the video and have a go. Alright, bro. 3, 2, 1. Okay, the answer. So, as you can see, if you look at these really closely, so every second molecule on cellulose is flipped. So every 6th carbon on starch is the right side up. As you can see, they're all pointing upwards. Whereas if you look at starch, every second molecule, so the second and the 4th, the 6th carbon is pointing downwards, as you can see. So they're clearly inverted. And the reason for this is because they are both made out of different isomers of glucose. So starch is made of alpha glucose and cellulose is made out of beta glucose. So remember, bro, this question isn't explained, so you need to say why. And so you get 1 mark for telling us that it's inverted, and you get the other mark for saying why. So the reason why is because they're both made out of different isomers. So if you did that, 2 marks in the tank. Come on, bro. Next question. Alright, in an damn! Bro, I cannot be... AQA really be telling stories out here. Alright, bro, let's get this one done. In an investigation, my bro wanted to identify the solutions in 2 beakers, A and B. My bro knew one of them had maltose and the other one has glucose. Both solutions had the same concentration. He did 2 separate biochemical tests on each sample. Test 1, he used Benedict's solution, and test 2, he broke it down. And then he used Benedict's solution. And it says here, maltose is hydrolyzed by maltase. And a quick little fact is, if something ends in ase, it's usually an enzyme. So I know some of you don't want to be reading bedtime stories right now, so I'm going to try and condense this little story here into a better, more digestible size. So it gives you the results here of bro. He did these 2 tests and it wants you to explain the results for beaker A and beaker B. So I'll give you a second to do this one. And yeah. So let's go through the answers together, bro. So we know off the bat that Benedict's solution cannot test for a non-reducing sugars, aka your disaccharides. So, in order to test for them, we must break them down in whatever means necessary. So we can use acid or an enzyme, whatever, right? And in this particular case, he used an enzyme. So, bro got the smarts. Now, so in the first beaker, he used Benedict's solution and he got red. So then he did it again, he broke it down and still got the same thing. So that tells me that there's probably not any disaccharides in there, right? He tested it, he broke it down, and the same color, it didn't change anything. So a thing about Benedict's test is that if you was to say, get a solution containing monosaccharides, and you test it, and you get, say, orange, right? So quite a bit in there. And you was to break it down and test it again. You cannot get a color that signifies less monosaccharides than orange. The reason being is there are already enough monosaccharides in there to give you orange, so there can't be any less. They don't just disappear magically. In fact, you could either get more or stay the same, if that makes sense, bro. Because if there were any non-reducing sugars, any disaccharides in there, you would be breaking them apart and adding more monosaccharides to your solution, therefore getting even like a stronger color change, so more than orange. However, if there aren't any disaccharides in there, it wouldn't change. And so this test signifies it perfectly. In A, he got red, which means a lot of monosaccharides. And then he broke it down with the enzyme and then tested it again, and he still got the same color. So no new monosaccharides have been added from the broken down disaccharides. Whereas in beaker B, he tested it with Benedict's solution, he got red, so a lot of monosaccharides. Then he tested it again, broke it, and he got an even darker red, so even more monosaccharides were added on. And there, the only place they came from was the already existing disaccharides in there. Right, so I know you didn't order a yappuccino, so let me chill. This is the mark scheme, so mark yourself accordingly, don't cheat. Did you get 2 marks, bro? Hopefully you understood. So, next question. Alright, bro. Here it says, the use of a colorimeter in this investigation would improve the repeatability of my bro's results. Give one reason why, and this is linked to the last question we just did, and that's for 1 mark. So, give you a second, get that one done, and we'll go through the answer. So remember, bro, a color change isn't good enough. It's not quantitative, so it doesn't exactly tell me what it is. And bro got red. Then yeah, red means there's a lot, but it doesn't exactly tell me how much glucose there is. So the best way I can analogize this is if I went to bro and asked, yo, what's the weather today, bro? And he said, oh, the sky is blue. Now, yeah, that does kind of tell me that the weather's good, right? If the sky's blue, the weather is probably nice, but it doesn't exactly tell me the exact temperature of that day. It doesn't tell me the forecast. It doesn't tell me, it doesn't exactly tell me enough. And so, yeah, if somebody told me, yeah, the sky is blue and clear, I'd probably have a very good idea of what the weather is that day, right? It's probably a nice day, but I wouldn't know if it's 15 degrees Celsius or 30. Okay, so I wouldn't know the exact temperature. And this links back with biology, right? Just because it's a red precipitate, it tells me that there's a lot of glucose, but it doesn't tell me the exact number, which is why we use a calorimeter to give us a value. So thereby quantifying our results, it makes the results quantitative instead of qualitative. It's just, if it's qualitative, it's more subjective, but if it's quantitative, it tells us an exact value. So if you got that, bro, or any of these 3 points, well done, bro, 1 mark. Alright, bro, it says here, my G carried out a Benedict's test. Suggest a method, other than using a calorimeter, that my G could use to measure the quantity of reducing sugar in a solution. Alright, I'll give you a second for this. Now, the answer. I went through this in one of my videos, but it's basically saying, how can I quantify my result without a calorimeter? And it's simple, bro. You just collect the precipitate and then weigh it on a scale. Alright, next question. Says here, yo, bro, explain how cellulose molecules are adapted for their function in plant cells. And this one's a nice, cheeky 3 marks. So I'll give you a moment. So this one's pretty simple. It's just basically, it wants you to just regurgitate everything you've memorized, and it really is that simple. These guys, they form long straight chains, and they form hydrogen bonds between each straight chain, and this forms fibrils which they link together. And this is really good because, like I said, these large quantity of hydrogen bonds on their own, they might not be that strong, but when there's so many of them, they are very strong, and they provide structure and support for the cell. So, these 3 points, 1 mark for each point. How much did you get, bro? Now, next question. Alright, bro. This one's giving you a little bit of a giggle. It says, tired yet, bro? Ha ha ha. Give me 2 ways in which the structure of starch is similar to cellulose. Now, there is so much you can say about this, because these guys have a lot in common, right? And a lot different. And so what you can say is, they are both polymers, right? They're both made of many, many, many glucose molecules. You could say that they both contain the same atoms, right? Carbon, hydrogen, and oxygen. Or like I said, it's in the name, carbohydrate, carbon, hydrogen, and oxygen. You could say they both contain glycosidic bonds in a 1 to 4 structure. You could say, they are both unbranched. Remember, amylose of starch is unbranched. Amylopectin is branched, but we can just ignore that for a second. Amylose is unbranched, just like cellulose. There's literally so much you could have said here, bro. This is like your kitchen. You can just cook. So, this is the mark scheme. If you got any 2 of these points, that's 2 cheeky marks in the bag, bro. Well done. Now, this one kind of roasts you a little bit. It says here, yo, you nerd. Explain 2 properties that make water important for living organisms. So, I'll give you a cheeky little minute to get this one done. So just like the last one, you could have said so many things about this, so many. But 2 is fine. You could say because of cohesion, it allows the water to go up a plant. You could say it resists rapid temperature change due to the hydrogen bonds, therefore it acts as a good habitat for, aquatic life, right? Because it doesn't change the temperature very rapidly, the organisms can stay at a relatively stable temperature. So here's the mark scheme. Could have said any one of these points, and if you said any 2, that's 2 marks, bro. Come on. Alright, bro. This one says, my bro, describe 2 differences between the structure of cellulose and glycogen. Do not include politics in your answer. So I'll give you a second again, and yeah. So just like the one comparing cellulose and starch, this one's very similar, okay? Because you got glycogen, a very branched polysaccharide made of alpha glucose, and you got cellulose, a very straight chain made of beta glucose. So there's your 2 differences here, but this is the mark scheme, and yeah, honestly, very easy. Let's move on to the next one, bro. Alright, this one says, my other G wanted to produce a dilution series of a maltose solution, so he could plot a calibration curve. He had a stock solution of maltose of concentration 0.6 moles per decimeter cubed, and a distilled water. He made a series of dilutions from 0.1 to 0.6 moles per decimeter cubed. Look here, my G's absolutely clueless. Can you fill out the table for him in it? So, have a go at this one, bro, and I'll explain the answers in just a minute. So the first part, concentration of maltose. It's basically just asking you what it's measured in, right? The first box, so generally speaking, when we measure concentration, it's usually always in moles per decimeter cubed. And it kind of tells you this in the question. So first box is pretty easy. And this box here is also pretty easy. So this guy's friend basically wants to dilute his maltose solution. It's kind of like adding water into your fizzy drink, right? You dilute it because it doesn't taste as fizzy, right? It's a bit watered down. This is what he kind of wants to do with maltose. And so it says in the question that he wants to dilute it with water, distilled water. So we put distilled water in this box, and it's obviously measured in centimeters cubed, because generally speaking, we measure the volume of things like water or maltose solution. We generally measure the volume of things in centimeters cubed. Now for the difficult part, calculating the concentration of maltose solution after you've diluted it with water. So this isn't exactly testing your knowledge on carbohydrates. This is more testing your knowledge on your math skills. So I'm going to go through with it and explain it to you. Say if I was to have like some kind of solution, right? I don't know, some kind of, goose juice, okay? I've had goose juice. I don't know what that is, but I just made it up. And it was, say, at a concentration of about 2 moles per decimeter cubed. And I had about 10 centimeters cubed of this solution, okay? So 10 centimeters cubed of goose juice, and I add 10 centimeters cubed of water onto it, right? So I add itself plus itself again, but in the form of water, okay? I hope you're following, but basically, if I add 10 centimeters cubed of water onto it, right, so kind of like, in a way, doubling it, but in water form, that would halve the concentration, right? So in this case, we have 5 centimeters cubed of maltose, and we add 10 centimeters cubed of water. So you haven't just doubled it, you've tripled it, because you've added 5 once, so itself once, and then again. So you've watered it down by a lot. And so the dilution factor, that's what we call it here, the dilution factor is 3. So maltose ends up getting very diluted. So I just start off by filling up the bollocks. We got moles per decimeter cubed here. That's what maltose concentration is measured in. Then we've got the volume of water, and that is measured in centimeters cubed. So, got that out of the way, now this part. So we use this little silly equation. It goes M1 times V1 equals M2 times V2. So, what does this mean, bro? So what do all these silly little letters mean? Well, M is simply the molar concentration. And V is simply the volume. Now, what does 1 and 2 mean? Well, 1 is the initial, right? The beginning, the starting version. Whereas 2 is the desired. So in order to use this equation, right, we just use a bit of quick maths. So we want to find the molar concentration at the end, right, the desired. So in order to find this, we just make M2 the subject of the equation. So in order to do that, we just divide V2 from both sides as we go. And then this is our new equation now. We have M1 times V1 over V2. And this should give us our concentration. So we plug all our stuff in. M1, the starting concentration is 0.6. Our starting volume is going to be 5 centimeters cubed. So what we started with, and our desired, our final volume, which is just added together, 15 centimeters cubed, 5 plus 10. And this, plug it all into the calculator, you get your answer 0.2. I would advise learning this equation, bro, because these dilution things come up so much in the exam. So M stands for concentration and V stands for volume, right? And the 1 symbolizes like the first part of it, right? The beginning. So if I'm talking about M1, I'm talking about the concentration at the beginning. If I'm talking about V1, I'm talking about the volume at the beginning. If I'm talking about M2, I'm talking about the concentration at the end. So I'm pretty sure you can get how this works. If you do want me to go over this a bit more, then just comment down below. Alright, bro, did you get it right? Yes, no, maybe? Alright, next one, bro. Alright, this one says, yo, my G, I got a date coming up. She's bad as fuck. Can you describe the structure of glycogen? So, if you do actually have a date, right? Do not talk about biology, because then she's just going to leave you on red. She's going to ghost you. So just chill out on, bro. Leave the biology, alone, okay? I can't even let the host know about this YouTube channel, otherwise I'm finished. It's over for me. Anyways, this one is pretty easy. All you have to do is just regurgitate the information that you've already memorized. Glycogen, very branched, big polysaccharide made out of alpha glucose molecules. Boom, that's it. Easy, 2 marks. Next question, bro. Alright then. This one says, got a really weird one for you, bro. Monosaccharides and disaccharides taste sweet. The lactose-free milk made after hydrolysis with lactase tastes sweeter than the cow's milk containing lactose. Suggest why. Alright, have a go at this one, bro, and I'll go through the answers in a second. Alright, this is a little bit of a weird one, I'm not gonna lie, because I'm not about that mummy milky business. If you are, then I don't judge, but I ain't about that life. Anyways, this one is not that bad, actually. Alright, so we know that monosaccharides and disaccharides both taste sweet, right? And let's say, for example, we had 5 disaccharides, and we broke all of them down. How many monosaccharides would we have? We would have 10, right? It only makes sense. So, if you had 5 disaccharides to begin with, and let's say the sweetness level was 5, right? Because each disaccharide contributes to 1 sweetness level. Again, this is just an example, just so I can make you understand. And we broke all 5 disaccharides into their constituent parts. So we broke them into monosaccharides. Now we have 10 monosaccharides. And now the sweetness level is even higher, because it's a level 10 now. And due to that, we have a higher concentration of sugars, which means more sweetness. So this can be true for this cow, okay? it has milk and it broke the lactose down into their constituent parts, which means more monosaccharides, more glucose and galactose, therefore sweeter. So this is the mark scheme. Did you get it right? Yes, no, maybe? Now let's do the next one, bro. Alright, this one's lovely. This one actually threatens you. So it goes, alright, bro, name the monomers from which a maltose molecule is made, or your ass is grass. So I am kind of shivering in my boots, but let's go through the answer together. So the answer is just 2 glucose molecules. I did go through this before. Just a bit of memorization. You better have answered, otherwise your ass is grass. Alright, next one, bro. Alright, last one then. This one says, explain how you would use the graph to determine the maltose concentration with a light absorbance of 0.45 arbitrary units. So I'll give you a second to do this one, and I'll go through the answer in a second. Alright, so this is pretty simple. This graph is showing us a positive correlation of how the concentration of maltose changes with the light absorbance. So the more concentration of maltose means the more light absorbed. So we just plot a graph of best fit. So let me try. Damn! Hold on, let me try again. Hold on. Alright. Okay. Damn! Okay, let me try again. Come on, lock in, let's go. Alright, perfect, that was amazing. Alright, now we plot here, 0.45, and we go across, and then we go all the way down here, and there we go, that's our concentration, 0.45. So, yeah, pretty easy, bro. It's just line of best fit, go across and down, and we got 0.45 moles per decimeter cubed. Easy, bro. Alright then, bro. So it's the end of the test now. Get your marks. So 65% is a grade A star. So that's 20 marks. 52% is 15 marks, which is a grade A. If you got 43% or 13 marks, that's a grade B. If you got 39%, which is 11 marks, you get a grade C. If you got 27% or 8 marks, you got a grade D. And I ain't gonna lie, bro, if you got this, you are cooked. Like, beyond cooked. So, tell me what you got in the comments below. And yeah, that's the end of the video. Make sure to subscribe the video, like all that bollocks, and yeah, I'll see you soon, bro.